Hi and a very good day everyone. My name is Nurin Binti Haminullah, student ID 20231049979 from AS244 Biomolecular Science. Today I will be presenting my final year project titled Identification of Manganese Peroxidase and Protease enzyme in Aspergillus niger and Aspergillus versicolor. Fungi are classified as grass organisms, which stands for generally recognized as safe. These organisms are highly appreciated for their ability to produce useful enzymes for various industries including food, pharmaceutical and environmental sectors. Among the many genera of fungi, Aspergillus is one of the most prominent due to its capacity to produce a wide range of extracellular enzymes, including lignolytic and proteolytic enzymes, which help in breaking down complex molecules. Specifically, Aspergillus niger is a well-studied species known for secreting large amounts of enzyme and organic acids. It is adaptable to various environmental conditions because it thrives within a wide pH range from 1.9 to 9.8. This versatility makes it ideal for industrial enzyme production, including MNP and protease. On the other hand, Aspergillus versicolor is commonly found in natural environments such as soil and in food product. It is less studied, but it is known to produce proteolytic enzymes and steric which is a carcinogenic compound. Furthermore, it is also known as an opportunistic pathogen that can cause infections in humans and animals. This study focuses on two important enzymes, which are manganese peroxidase MNP and protease. These enzymes enable the breakdown of complex substrates. Literature focuses on wide root fungi such as phenylocate chrysosporium, while studies on Aspergillus are limited. There's also lack of comparative data on how Aspergillus niger and Aspergillus versicolor perform under specific enzyme-inducing conditions like manganese sulfate MnSO4 for MNPs. This gap in knowledge limits our ability to optimize enzyme production from this species for large-scale industrial use. This study is significant because it investigates the enzymatic activity of MNP and protease in Aspergillus niger and Aspergillus versicolor. While MNP is typically associated with wide root fungi, its presence and role in Aspergillus species, especially Aspergillus versicolor, is still underexplored. By studying these enzymes in both species, this research contributes new insight into their biotechnological potential, particularly for industrial enzyme production. This research was conducted to address the following questions. 1. Does Aspergillus niger produce MNP and protease enzyme? 2. Does Aspergillus versicolor produce MNP and protease enzyme? Objectives of this project are: first, to qualitatively determine the presence of MNP and protease production by Aspergillus niger and Aspergillus versicolor on different agar media. 2. To quantitatively determine MNP and protease activities using UV visible spectrophotometric analysis. Study utilized several media such as PDA, potato dextrose agar, potato dextrose work, PDB, Cesapex dot agar CDA, and skim milk agar SMA. All media were sterilized at 1 to 1 degrees Celsius for 15 minutes to ensure no contamination. Fungus spore suspension revived on PDA. A spore suspension was prepared using 0.1% twin 80 and quantified using a hemocytometer to standardize the spore concentration. Fermentation, the MNP assay was conducted by culturing fungi in PDB supplemented with 0.033% MnSO4 and 3 ml standardized spore suspension. Protease assay used PDB with 3 ml standardized spore suspension. The qualitative assay, MNP activity was identified by color change on CDA from red to yellow using phenol. Protease activity was identified by the formation of clear zones or zone of hydrolysis on SMA. Quantitative assay, MNP was measured at 610 nanometer using phenol as the substrate, and for protease activity was measured at 660 nanometer using tyrosine release measured via folding reagent. For qualitative analysis, CDA plates containing phenol were used. A niger exhibited a less intense yellow color indicating lower MNP activity. A versicolor showed a more intense yellow color indicating higher MNP activity. B pulmonaris, which served as the positive control, demonstrated the strongest color change. Negative control quadrant remained red, indicating no MNP activity. Color change from red to yellow occurs due to acid byproduct formation from MNP activity, which causes a drop in pH and activates phenol red color transition. Species can produce MNP with Aspergillus versicolor exhibiting stronger activity. This activity was screened using SMA plates. The formation of clear zones around the fungal colonies indicated protease secretion. A niger formed clear zone confirming protease activity. A versicolor also formed a clear zone confirming protease activity. The chrysogenome or the positive control showed the largest clear zone, and the negative control did not show any clear zone. Both A niger and A versicolor can effectively secrete extracellular proteases, supporting their role as potential industrial protease producer. This activity was screened using quantitative analysis. The MNP activity was measured using a UV visible spectrophotometer at 160 nanometer over a period of 4 minutes. Urban increased as follows: Aspergillus niger from 0.729 to 0.742, Aspergillus versicolor from 0.757 to 0.769, and pulmonaris from 0.909 to 0.976. According to figure 3, all species display a linear trend in absorbance increase with R-squared values above 0.91. Pulmonaris had the fastest rate of change with an R-squared value of 0.9784, indicating the strongest MNP activity among the species. Between the two Aspergillus species, A versicolor showed higher MNP activity than A niger. Versicolor produced a more intense yellow color due to faster acid byproduct formation, which lowered the pH and causes a more significant shift in the phenol rate indicator. The activity of MMP in units per milliliter from 0 to 4 minutes for A niger, A versicolor, and P pulmonaris. At 1 minute, MMP activities were for P pulmonaris 786.36 U per mil, A versicolor 304.55 U per mil, and A niger 150.00 U per mil. As illustrated in figure 4, the activity decreased over time for all three species due to substrate depletion or enzyme inactivation as the reaction progressed. Among the three species, P pulmonaris had the highest MMP activity, followed by A versicolor, and lastly A niger. A versicolor showed a more intense color change on CDA plates, which is consistent with this data. These results are supported by Westenberg, who reported that Aspergillus species generally show low MMP activity. However, the current method showed reach high efficiency as the MMP value of P pulmonaris at 786.36 U per mil far exceeds the 70.20 U per mil reported by Martin. The quantitative analysis of protease activity was determined by measuring casein hydrolysis at 660 nanometer using the following reagent. The amount of tyrosine released from the hydrolysis was measured using a standard curve as shown in table 2. In figure 5, the standard curve was plotted using different concentrations of tyrosine. Linear regression equation obtained was y equal to 0.0038x plus 0.0216 with a correlation coefficient of r squared 0.9586, showing a strong correlation between tyrosine concentration and absorbance. Based on table 3, the absorbance values of different fungal species were for P chrysogenum 0.717, A niger 0.668, and A versicolor 0.665. Both A niger and A versicolor successfully produced protease enzyme as they showed a significant absorbance at 660 nanometer, slightly below the positive control. Protease activity measured in units per milliliter, niger showed 102.03 U per mil, A versicolor had 101.59 U per mil, and the positive control P. chrysogenum recorded at the highest value at 109.80 U per mil. Before show you the protease activity value with the representation of bar chart, strong correlation was observed between absorbent values and the calculated protease activity across all species, which confirms the reliability of the spectrophotometric method used in this study. These findings are consistent with Zaidah, who reported that Aspergillus species can produce high levels of protease under appropriate conditions. Overall, A niger and A versicolor demonstrate comparable activity to P chrysogenum, supporting their potential as industrial protease producer. To conclude this presentation, both Aspergillus niger and Aspergillus versicolor successfully demonstrated enzymatic activity for both MMP and protease enzyme. MMP activity was confirmed using the phenol rate decolorization method. The red color changed to yellow, especially around the A versicolor, indicating stronger MMP activity than A niger. For protease, clear zones formed on skin agar, confirming protease activity in both species. Quantitative analysis, MMP activity was measured 610 nanometers, as A versicolor recorded an activity of 304.55 unit per mil, while A niger showed 150 unit per mil. P pulmonaris had the highest activity at 786.36 unit per mil. For protease activity, casein hydrolysis was measured at 660 nanometers. A niger produced 102.03 U per mil, while A versicolor produced 101.59 U per mil. P chrysogenum showed the highest activity at 109.8 U per mil. These findings confirm that both A niger and A versicolor have excellent potential for industrial enzyme production. Their activities are suitable for biotechnological applications such as bioremediation. For future directions, I recommend optimizing culture conditions including media composition, pH, temperature, and metal ion supplementation such as manganese. Additionally, using alternative substrates may further enhance enzyme production, especially for protease. Thank you very much. That concludes my presentation.